Come on, Argus, you can do it. Bridge nav, 20 meters, same bearing.
Another CP clamshell.
Yeah.
You see how that goes? Carbonate rocks here?
These do look like carbonate.
Yeah. Science, as we kind of go along these contours, do you want me to, the angle of the slope has changed. Do you want me to kind of face the slope a bit more and change my heading, or would you rather maintain this one as is?
Give me a second to see if Tom chimes in, since he's on a little bit of a time delay. He says either is fine. So why don't we turn in a little just so we get a little more of the view
Roger.
in the light pool.
Roger that. Bridge nav, another step, same bearing.
Yeah, he says whichever allows for good view and safe flying.
All right, let's try that one there.
Nice Tanner crab.
Seeing all the tuna pools in the water column reminds me that in 2017, when the Nautilus was here last, it was in the middle of this pyrosome bloom, where sometimes they were so thick during the whole descent and piled up on the bottom. Different kind of pelagic tunicate.
And there were piles of them.
Yeah, and every anemone seemed to be feeding on one.
Mm-hmm. Haven't seen many of on this dive so far as the deep sea jelly. Lots of ctenophores, but not any of the jelly.
No, it's true.
I'm sure they're around. Anemone there that's all closed up.
No.
Well, there's a kind of thin structure coming down that slope. What looks like wood carbonate.
Bridge nav, another step, same bearing.
Open to mud.
And we're back out into the mud. Seems to be the luck of the transects today, although we
Sure
did get
does.
some corals on this one.
Yeah, it wasn't a bad one.
See, what we're learning is that the corals seem to be spaced about every 200 meters.
It was better for sampling this way around.
Bridge nav. I have another 20-meter step, 005. Thank you. Thank you.
Tom has been watching back the video on the stream and said that the eel pout that we zoomed in on a while ago may have been a Kamchatka eel pout. That it's similar to the snakehead in the same genus, but without any of the shiny scales.
Hooray! What the zoom.
For anyone watching at home, they do have on the main Nautilus Live channel the ability to kind of rewind time and watch things.
Those
If
more
you
There. Doesn't really look like it.
Okay.
Looks more like a little rock with ophiuroid arms sticking out of it.
Very
Yeah,
interesting.
I think so.
ophiuroid with only 3 of the arms sticking out.
What was it? Just
I don't at the bottom of the screen yet. It may also be missing arms if they're not buried, but they can regenerate them.
We're almost 200 meters. And I think just in preparation for handing over, I'm going to have a step out directly to the east 10 meters because the
Okay.
wall's starting to curve back in. I don't want it to fall in.
Yeah.
Bridge nav. Can we get a 10-meter step bearing 090? Thank you.
Hello, Stephen.
Yep.
So I think we got a pretty solid 200 meters in there. You happy to
Yep.
call it end of
Sounds good.
transect?
Yeah, go ahead and call in transect.
All right, end of transect. Good timing.
And Tom has chimed in from shore to say great transect, and he'll be rejoining later this morning.
Thanks, Tom.
So for the next watch that's coming on, where will they be continuing on to? Maybe I can get them set up so that they're in a good position to just get going.
Yeah, I think the next waypoint, I'm trying to bring up the camera 3,
Zoom in
the view
out.
3.
So it's
Is
up
that number 1? Okay.
1.08 is at a ship bearing of 032. Okay. That's about 350 meters away. Okay.
Yeah, I'd say let's start heading up towards that. It looks like we're gonna gain some elevation there, and kind of get into a new depth band. So yeah, just I would start the ship moving up kind of towards that direction.
Okay. And this is going to be in exploratory mode, correct?
Yep, this is an exploratory mode.
All right, great. It's going to be definite up contours to go direct to that. Comfortable to do that? Yep, that's fine. All right, so I'm going to do a direct 032. 032, roger. Very small steps, 10 meters. Sure. Bridge nav. Can we get a step 032, 10 meters?
For those who are watching at home, we've just finished doing one of our quantitative transects, and we're now, the pilot and the navigator are getting the ship moving up towards our next waypoint. We've relayed on this. As we get ready to hand off for the next watch in 5 or 6 minutes.
Going to be getting like 100 meters of vertical change over that. Right.
Yeah, so we've been moving along slope, and we're going to be moving more upslope as we move forward.
I'll make note of that, Bob.
Big boulders to finish.
Nice percentage star.
Huge percentage.
Bridge nav, another step, same bearing. Kim, would you mind bringing your heading to 032? Thank you. Bridge nav, another step, same bearing.
Yep. We seem to have just lost the data feeds on the science screen remotely.
Yeah, mine's been failing on the chat for a while. Oh, glass sponge. Big anemone.
You take a steep, muddy, with just a little hard substrate underneath material.
It's changing, watch.
I can hear you. In my headset, it sounds fine. It's been fun. I say, poor Tom, we haven't even been like trying to get the transects in the mud. That's just where they've been happening.
At least you got the last
All
one.
right.
That was interesting.
Yep. So for the watch change, we just ended a transect about 10 minutes ago. I don't think we've made that much forward movement. So you'll be in exploratory mode for another 100 meters or so before they're ready for the next one. And with that, I'm going to click off of my SPL and turn things over to Jenny for the next shift.
Me too. Thank you very much, everybody.
Yep,
Great
thanks.
samples. Good dive.
See you in the science chat for a little while.
For those of you just joining us, we're in the middle of a shift change at the moment. We're going to be changing out the staff on board the ship as well as the scientists ashore who are helping to guide our efforts. We'll be starting another 4-hour shift here soon, so just hang in there and feel free to check out the recent video while you're waiting. Just wanted to share that this collaboration between NOAA and the Ocean Exploration Trust is really quite an amazing opportunity for us to showcase unique science and education capabilities of the EV Nautilus by exploring and studying within national marine sanctuaries and some of the adjacent important ecological areas that we all care about. Really the live telepresence capabilities of the Nautilus are pretty unique, and it allows scientists from all around the world to participate and collaborate and contribute to the success of this mission. There are quite a few fantastic tools that Nautilus and Ocean Exploration Trust has developed, some recently developed to help enable us to actually pull off a dive like this with uh, deep sea exploration safely from a ship, as well as engagement of scientists ashore like me and many other scientists who are working on this expedition. We're very pleased to be able to bring the ocean and ocean science to you all over the world, as well as to classrooms and students across the planet. This is a pretty amazing opportunity. The Nautilus is an incredible vessel. It's about 64 meters long or 210 feet. It's operated by Ocean Exploration Trust and has been a real asset to us on the West Coast here for exploring our deep coral and sponge and seep ecosystems, as well as the other representative habitats that we have in Olympic Coast National Marine Sanctuaries, as well as outside areas. I'm excited to let you know that we are just the first of several marine sanctuaries on the West Coast that will be featured in the expedition season this year. We're going to have colleagues of ours joining a little bit later in the expedition season to do some great exploration off of California. And I invite you to tune in for those exciting expeditions. In many ways, we are all linked through an initiative that's happening right now called the West Coast Deep Sea Coral Initiative. It is sponsored by NOAA's Deep Sea Coral Research and Technology Program, and a lot of the funding for the video analysis of transects and the species identification and taxonomy, the eDNA work, and many other products are made possible through collaborations between national marine sanctuaries, the Deep Sea Coral Research and Technology Program, and our partners like Ocean Exploration Trust, because they have the capabilities that allow us to do all this great work. And we're incredibly grateful and feeling very fortunate, especially when we have dives like we've had this morning, where we've identified and located some really inspiring places. So please continue to join us. We are getting our watch kicked off. I'm wondering, are all the ROV pilots in place? Do we have Sci 2? Do we have data logger?
Yeah, hi Jenny, this is Steven, data logger. Morning.
Good morning, Steve. I'm so glad you are with us for this portion because of your expertise with invertebrates. Please chime in as you see fit. Who else have we got? Hi, Nicole. Good morning.
Dave in the video position.
Hi, Dave. Good morning.
Yeah, can I get a ship move bearing 030 10 meters again? Yep, thanks. I saw some big ctenophores on the camera.
When the ROV pilots are ready, could you introduce yourself, please?
And I'm Ben Gracian, and I'm the navigator for this watch.
Thank you, Ben. Thank you for all your nav.
No problem.
Ben, can you see on the Norbit or any of the tools you have the
Can we have a ship
escarpment
move
that seems to
bearing
be?
030 10 meters? I actually don't think we have the Norbit sonar.
Yeah, we don't have Norbit up here
right now. I'm seeing just a lot of particulates in the camera. But yeah, we're on this relatively steep slope, so I'm just trying to keep along the same contour and slowly creep up upslope towards the next target. So we're looking at our sonar on the ROV to kind of make sure that what we're seeing with our navigation solution is matching where the cliff actually is, where the slope is. Because we want to make sure that we're not bumping into anything. On the way over, we were able to do a multi-beam map, and we had existing mapping data, and they lined up really well. So we use the mapping data we collect to aid us in our navigation.
That's great. I'm glad to hear you were able to accomplish that prior to the dive. Thank you.
Yep, great. Bridge nav, can I have another ship move? Same bearing, 10 meters. Great, thank you. I'm seeing a lot of these like low bait ctenophores. I think those. It's hard because there's this other species or genus or group, Boroidia, that looks kind of like that, like a thimble shape with these long comb rows along the side. And they're both predators, but the one eats the other.
Yeah.
Nav, would you please let us know when we're about 100 meters from the end of the previous transect, please?
Yeah, that's actually right now. We're about 110 meters from the end of that last transect.
Thank you.
And so you're going to want to start another transect a certain minimum distance away from the previous one? Is that?
Yeah, we need to have them spaced at least 100 meters apart.
Roger.
But so that's why I was asking, but I'm not ready to start one just yet. But thank you.
I'm in
Good to know.
no hurry.
Are we still marching along?
Yep.
Yeah, why don't we just keep marching along, and yeah, thank you for panning up a little bit. We'll keep following along and identify, if possible, the escarpment on the left and look for outcroppings that may provide the substrate necessary for the organisms that tend to like hard bottom and
Okay.
fixing to the seafloor. Nice to be in exploratory mode for a little while and see what we see. We've seen several of these grenadier fish over the course of the dive. Interesting deep sea species. Ooh, interesting sponge.
Did you want to zoom
Could
on
we
it?
please get a, yeah, let's please get a
Zoom in,
zoom on
Dave?
that one if you could, please.
I think that might be a closed anemone.
Oh yeah, it is. Maybe we've made it close up, do you think?
Doesn't seem
I'm
unlikely.
Could be many
scared it.
reasons. They they do that when they digest a meal too sometimes.
And are they feeding on particulates, or are they going for bigger items?
Whatever falls into their grasp
Yeah,
for the most part, but yeah, larger plankton, particulate matter.
easy living.
Hey, good.
Yep, let's go on. Thank you.
Bridge nav, can I have a ship move same bearing 10 meters? Yeah, can I have a ship move bearing 030, 10 meters? Yep, great, thank you.
For folks who are just joining us, we are in an area called an essential fish habitat area. It is called Biogenic 2 EFH. It is just west of Olympic Coast National Marine Sanctuary, and part of it lies within the usual and accustomed fishing area of the Quinault Indian Nation. We're going to provide a little bit of context for you in this next watch to help viewers understand where we are and why we're here exploring.
Yeah.
There's a look at the team in the van.
Is that a flounder of
Here
sorts?
we are
You wanna
coming
zoom on him, Dale?
across. Is that a deep sea sole and a tanner crab?
Looks like a standoff.
Beautiful. Thank you for the zoom. Watch this
Yep.
guy.
Looks like it has an isopod on it or something on its dorsal side.
Oh
It's
yeah.
Oh,
a
yeah.
small critter.
That white mass thing.
Yeah, about midway down the body, there's a
Hitchhiker.
parasite.
Could you zoom out a little, please?
Mhm.
And he stops moving. Yeah, thank you.
Good.
Yeah, thank you.
I
Let's
could
see
do
what
that.
else we got here.
Yeah.
Oh, there's another one. Maybe there's a whole pile of them here.
That's a fun thing. We get to find out. Is that one of the Paralia jellies that we see?
That dark jelly.
Yeah, the dark bell-shaped.
What is a Paralia? Is it a ctenophore or like a medusa? It looks like a medusa.
Steve, you want to jump in here?
Yeah, they're
You want to zoom in, Dave?
large planktonic organisms. This jellyfish, yeah, it is a jelly.
Medusa.
It's
Yeah, we'll do a creature feature. We'll see a lot of these guys probably over the course of the dive. Ooh,
So
they get pretty large, but and this one's average size. There's been years in the past where they have just been so abundant, you can't get them out of the camera
thick.
view.
Mm-hmm.
They're pretty opaque too.
It's also seasonal, so you know we are diving much later in the year this year, September.
Yep.
All right, I'll put
Okay, let's
another ship.
continue our ramble on, if we may, please. Yeah, I'm noticing the absence of the pyrosomes that were so abundant during our 2017 cruise. Steve and I were able to work together in 2017, we had a 17-day Nautilus cruise within Olympic Coast National Marine Sanctuary where we were able to use both ROV Hercules and an autonomous underwater vehicle called Popoki from NOAA's Northwest Fisheries Science Center. And so we were able to pair the AUV dives in the day with deep ROV dives in the nighttime. Did a little mapping as well, but it was an incredible opportunity. So we're excited to be exploring more areas that haven't been visited before now that we have Nautilus available. This is one of the few tools we have to visit deep areas of this coastline, this Olympic coast, which is, as you can see, an interesting place with a diversity of deep sea habitats. So, this morning we are exploring an escarpment in biogenic 2 EFH, just west of the sanctuary boundary, and looking for deep sea coral habitats as well as seep habitats and other deep habitats that are new to us. So thank you for following along, everyone at home. We're excited to take you along with us on this little journey of exploration. I think let's try and start a transect in the next 5 minutes or so. So let's get set up for that and try and find a 200-meter stretch if we can that we can survey using the quantitative transect protocol that's being used all up and down the West Coast. We'll be basically moving along the seafloor at a relatively stable altitude for approximately 15 to 20 minutes in an attempt to cover about 150 to 250 meters of seafloor. We're going to be running the camera and the lasers that you can see on film that are about 10 centimeters apart, or exactly 10 centimeters apart, which allows us to size organisms like this probably long spine thorny head, which are the smaller and deeper species of thorny head that we have in this region. Someone want to jump in with a suggestion?
So we're on a little bit of outcrop, just a slight outcrop. So for the next 50 meters, so I can keep a straight line for 200 meters, but it's not going to be perfectly at this fixed contour. Oh, you want a fixed altitude?
Why don't we?
You don't care if we're going up slope?
Let's get past the outcrop. Let's get past the outcrop and then start a transect. Will that work?
Yes. And so, again, you don't need it to be at the same depth. You just need us to be at the same altitude?
Yes. It's fine to do a lateral ascent as we're moving through the transect.
No, I was talking to myself. Sorry. Another one of the souls, right?
As you can tell, the substrate here is very silty and muddy. So pilots, if you could come off the bottom a little bit more to try and avoid that suspension, that would be great. Thank you. How are we doing making it around the to where we can start a transect? Are we waiting on the ship?
Yeah, we're like 40 meters from where it becomes really flat, but I don't think it's a problem to start here either way. It's getting more and more gradual as we come along it.
How does the habitat look off the port side? Are we looking at steeper habitats on the port side that we can ascend?
Yeah, so the slope is at an angle of like 350 degrees, if that makes sense, or like a little off from north.
Uh-huh.
Or actually, maybe more like, maybe closer to 330. So yeah, to port will be our upslope view.
All right, well, when you are set and ready to start a transect, let's move through this cloud and hit a start transect on the data logger and move through our first transect for this watch, please.
Okay, roger.
Thank you. We're going to be trying to move at about a speed of 1/4 to 1/2 knot over the seafloor, with fairly consistent speed and altitude for about 15 minutes. So we're not going to be in exploratory mode, stopping and doing a lot of sampling or checking things out, but we will talk with the video engineer to make sure we have wide field of view and are able to identify the species that we see. Hoping that the science chat will be up again soon and that Tom Leidig, whose team analyzes these transects, will be able to join the fun.
Yeah, whenever you guys are ready, I'll just mark.
I'm ready.
Okay.
I don't want to hang around.
Yeah, yeah.
The cloud's
So
kind of
if we're on a
following us.
fixed altitude, I'll just mark this as the start.
Yeah.
And I'll keep
Moving
it.
along is good.
Yep.
Video's
Yep,
all ready.
let's go.
We're zoomed wide and at far focus, so we're ready to go.
Bridge nav.
All right, let's go, please. Some questions for the ROV pilots as we go. How easy is it to maintain speed over ground?
Well, I'm somewhat limited because the ship kind of moves in stagger steps because we don't want to get, depending on how steep the terrain is, you don't want to get the ship too far out, you know, moving too fast along the bottom. You don't want to step too far in case the cliff comes up at you.
You're
'Cause
highlighting Ben's role as the navigator in communicating with the ship, right? And making sure that we're all coordinated with Argus, Hercules, and Nautilus. It's quite an intricate dance for those of you following along.
That white thing, another clamshell up there?
Looks
like it. Yeah, I'm just making verbal annotations for the shore. It looks like a clamshell, maybe.
Yeah, it does appear to be the species that Andrew gets excited about. It's a solo, though, so we have fallen from higher up. Or pure speculation on my part.
I think that's sound speculation until clams can start to climb from deeper depths.
All right, let's keep moving to stay out of this disturbed sediment plume if we can, please. You know, it's hard to avoid in these very silty habitats down here.
Are you still doing steps, or you?
I'm just gonna do steps consistently if that's cool. I'm doing 20-meter moves though.
With respect to sampling, I know that we have filled our method or our push cores, I believe. Is the quiver full
We
for
have
push cores?
3
Okay,
of 5 push cores taken.
thank you. And how many bio boxes are currently empty?
The majority of them, we have 5 bins open and most of the flush jars as well. Most of the slurp jars as well.
Oh man.
Thank you, Steve.
And just for good measure, just 1 Niskin so far.
Ah, thank you. Yeah, always good to check in. We have targets for the sampling that we'd like to do
Can we
for deep
march
sea
quicker?
corals and yeah, please
we
do.
just
Please
because
maintain.
we just the cloud's catching up to us
Yeah,
because we're not.
I mean, we
marching along.
haven't missed any steps.
Yeah, we're
want
just
me to
going
ask
really
them?
slow though.
Yeah,
We're
yeah.
definitely not doing quarter or half a knot.
What is that because of the ship? Because I would
It's
like to
just
go at
how fast the steps are happening because
all right, let's do a big step then if we can, please, so that we can actually do a transect.
yeah.
I'm wondering if we want to abort this one.
As far as the cloud goes, it's just the rocking of Herc just from getting tugged on a little bit's enough to disturb the bottom,
Yeah.
like barely thrusting at all. So, but as it gets yanked, it kind of sweeps the bottom.
Well, the ship moves have been coming in.
We're just not. I mean, I could see we're not traveling over the bottom very rapidly here.
All right, well, I think we may have to abandon this transect and just start over once we get back into a place where we can see. I don't have
Maybe it's
science
better to
chat
like
available.
just do a line at a set rate, Yeah.
Yeah, no, that's what I'd like to do.
Yeah, I feel
Set
safe over
speed, set
here, so
altitude.
I'm happy to track a line. And what speed would be the best for you guys?
Well, we're talking like 0.3 or something.
Okay, 0.3 knots.
Yeah, between a quarter knot and a half a knot is the target. So 0.3 would be great.
We want to do a continuous thing and not all these stagger steps are no good because this is really flocculent bottom here.
Thank you, Nicole. I'm going to try and get back into side chat.
Just to confirm with shoreside folks, did we abandon that transect or are we still continuing?
I would like to get out of this cloud and start over.
Okay.
I think I think we need to get everything in place with the ship and Argus before we start so that we don't get tugged so much, if that's possible.
Great. Just making a note of it.
Thank you, Steve. Nav, can you tell us when we're in good position to start another transect, please?
Really
I'm just going to confirm with the bridge that we're tracking a line, and once we're doing that, we should be all set.
Okay, we'll just go exploratory mode until you give us the go ahead to start.
Yeah, he just put it in, so we can start.
All right, let's try this again. Data, could you please start us on a transect? And
Yeah, you want to,
pilots, please
do you want to reuse
proceed at
5?
point. Sure.
Okay. So we're going to call this one transect 5 again.
Sounds good. And for folks at home, we are following a consistent protocol for
Not
transects
yet
that will
though.
be used throughout the expedition all the
I'll
way
get
down
more
through
so I'm in
California.
line. Yeah, I'm gonna get just out in front.
Yeah, that makes sense.
So yeah.
Yeah, our bearing is 025.
I don't want to get tugged at all because this is making it.
The vertical motion. Yeah.
Well, when you get a little tug on the tether, you get, the vehicle kind of
yanks up.
Well, it stirs up the bottom.
Right.
Like. And it doesn't take much in this stuff.
Yeah, it looks that way.
Okay. So now I'll start doing